Yeah.
Okay, cool. So we said we had 3 processes. This is the first one we're dealing with now. So this is the cell, and I mean, this is the nucleus is actually the nucleus. No, this is the cell, sorry. The cell, and then RNA polymerase is able to attach to a specific area. So RNA polymerase is the enzyme that produces the mRNA molecule. So first of all, we have to make it before we even start. So the mRNA molecule, the messenger RNA molecule, is made by RNA polymerase. And this process requires the DNA double helix to be partially unwound in the region of mRNA synthesis. So this is a pretty much intricate process. So everything has to be done very intricately and, specifically. So don't think that because it's unwound, it's going to be, detached and all of that ugly stuff. We're not trying to do all of that. We're just trying to get a little area so that we can attach the RNA polymerase so that we can essentially, develop a strand so that we can get our protein. I mean, we're not trying to go too crazy. We're just trying to copy from the DNA molecule, essentially. So that's basically how all cells work in dividing. They basically copy. They do the same process where they copy, and then they just mend it back together after a while, and then we're going to deal with it later, so you're going to understand afterwards. Everybody get it so far? Anything you want me to explain? Just let me know. Everybody good? And if y'all have any questions about the things previously, just, you could unmute your mic, you could, okay, cool. Everybody seems to be on a good page. Okay. Why isn't this moving? All right, so we're dealing with the promoter section of it. So there's going to be something called a promoter, where it's basically where everything starts. Okay, so the DNA sequence, I'm just trying to get my bearings with this part of the lecture. The DNA sequence onto which the enzymes bind to initiate the process is called a promoter. So this is where basically everything starts, right? And the promoter has important sequences. And these sequences would be basically the -10 Prinbo Tata box, which would be this specifically. So this is just something that you could commit to memory. You don't have to go too much in depth with it, but I mean, it's this is a classic thing that you're supposed to remember. So TAT, T-A-T-A, well, TATA actually. And then they have another one called the TTGACA. So this one, the other 35 bases after transcription of the start site. Okay, so this I am not too familiar with, so I'll get back to this. I think I'm gonna get back to that one. Okay, so the eukaryotes. The RNA polymerase requires the aid of several proteins for recruitment on the promoter. And these are special transcription factors which form the Basal Transcription Complex. Okay, so this is what we're essentially getting at. This is the promoter sequence here, and okay, yeah, so this is basically what we're starting with, the promoter sequence, where the mRNA polymerase would bind to so they could start the process, essentially. And these special transcription factors are that form the basal transcription complex. So yeah, that's basically what we're doing here. Oops. All right, so for this complex to be formed, acetylation of the histone tails is required to disrupt this association, leading to the weaker binding of the, sorry, I'm not seeing the chat. Oh, okay, no problem. No problem. All right, so this is just, we're dealing with eukaryotes. So basically for almost the entirety of this lecture, it's gonna be eukaryotes we're referring to because Yeah, that's the more complex one. So the RNA polymerase requires the aid of certain proteins for recruitment on the promoter. Certain proteins are required for the promotion, and that would be this area here, required for the promotion. And these are special transcription factors which form the basal transcription complex. So as I said before, Chris would have more knowledge on this because we kind of separated the lecture, but yeah, I kind of have a little bit of knowledge, but that's basically what this is saying is that for something to happen, it would require something else to be there. And the other things that would be required, which would be the certain proteins, which would be these certain things here, these particular things that are in this area. But don't worry, guys, y'all are gonna get it eventually. Hopefully Chris comes back. If he doesn't, then we'll just have to proceed. And either way, y'all are gonna get it in the end, I promise. All right, so for this complex to be formed, and the complex that we're talking about is what? The basal transcription complex. Basal transcription complex. So basically, the thing that is required to initiate this whole transcriptional process. All right, so for this complex to be formed, acetylation of histone tails is required to disrupt this association, leading to weaker binding of the nucleosomal components. Therefore, more transcription factors can reach the DNA. And yeah, so this is just something that you could also keep in mind. Yeah, so we're gonna get more in depth, don't worry. And once the RNA polymerase 2 can bind to the promoter, which would be, yeah, we should have put the picture on the inside. Yeah, so once it could bind to this promoter section, I know it looks a little complex, but it's a zoomed in representation of what's actually, you know, there. So the promoter, this is the promoter sequence, and we're trying to attach, sorry, my mouse. We're trying to attach the RNA polymerase 2, because remember, we're dealing with the 2, so just remember the RNA polymerase 2, and it's supposed to be attached here, but remember, for something to start, it has to have other things. So yeah, so now that we have all of those things, we can start the next phase of the process, which would be elongation. All right, so as we said, we have to open up this, this DNA strand. So we have a DNA strand, and it's the double-stranded DNA, because, you know, we have single-stranded DNA. We never really need to go too much into that, but it's the double-stranded that we're dealing with here. And then, okay, so the DNA is continuously unwound ahead of the core enzyme, which would be the mRNA polymerase 2, as the hydrogen bonds are broken. So we know that we have hydrogen bonds between the DNA molecule holding the bases together. So basically that would be broken temporarily. Let's just keep in mind that this whole thing in the end, we're still gonna have this DNA perfectly together, and, you know, in the ideal, because, you know, we always having some issue with some people, but in the ideal thing, we're gonna still have our DNA in the end of this process, but we're just using him. These people fake, they just want you for a little while and then they're gonna leave. But I mean, they're fake, because I mean, they still gonna, you know, make proteins and help the body, but yeah, y'all get the gist, right? I wanna make sure everybody's on the right page. Sorry, I'll make sure y'all on the right page. Oh, cool, got it. All right, sorry guys, I'm trying to manipulate this thing, and I'm young, but I have old mind. All right, okay, so the prokaryotes, yeah, prokaryotes. All right, yeah, so we're dealing with prokaryotes here, because you're dealing mostly with eukaryotes, but let's get into the prokaryotes aspect of it first. So the DNA, as I said before, is continuously unwound ahead of the core enzyme as the hydrogen bonds are broken, which is the bonds between the DNA molecule. The DNA is rewound behind the core, rewound, as the hydrogen bonds are reformed, because remember, we're still going to have an intact DNA here, but we're trying to use it so that we can get an mRNA, essentially. I didn't mention the mRNA too much, so I don't really want to mention it now until later in the slides, but the essential thing that we're trying to do for transcription is essentially to make the mRNA molecule, and that's what we're doing here, right? So we're still going to form back these bonds once it starts to elongate in this area from the 5' to the 3' section. Once it's elongated and we don't really need the things back here anymore, it's going to form the bonds again. All right, so we were dealing about prokaryotes before, now we're dealing about eukaryotes. All right, so the mRNA polymerase is released from the other transcription factors, and elongation can proceed as it does in prokaryotes, with the mRNA polymerase synthesizing pre-RNA in the 5' to 3' direction, right? Okay, so yeah, we're going to talk about this more. So keep this in mind, pre-RNA, pre means before. So we're trying to deal with RNA. We don't even know why they have this pre-RNA here, right? But we're dealing with this before, and you're going to see why they call it pre-RNA. All right, so for RNA synthesis to occur, the transcription machinery needs to move histones out of the way every time it encounters a nucleosome. And this is done by via FACT, which is it facilitates the chromatin transcription. All right, so for it needs to move out of the way. All right, so yeah, so we're just basically forming back this thing as the mRNA is being formed. It moves them out of the way and forms back the bonds and all of that. Okay, and this is the last process of transcription, and he has the prokaryotes thing here. Okay, so signaled by the P-independent mechanism comprising of GC palindrome, which forms the hairpin and about 6 uracil bases. In the DNA, okay, this is hair. The hair loop prevents
Alrighty. Guys, hi.
Yeah, we could hear you, Chris.
You hearing me now, Olivia?
Yeah. Pretty clear.
I am so sorry, guys, and I realize you guys have reached quite a long portion.
We haven't reached that far, Chris.
Yeah, long and short of it. I really hate digital, guys. I'm not going to hide it. So yes, I tried now by using data. Hopefully it will allow us to have a better reception. So yeah, I mean, and I think they hate me as well, because I have been filing complaints through the Consumers Affairs because of their rather unreliable services, and they're quite aware of me. So Olivia, you want me to, because I'm assuming.
Yeah, there's some sections where I could have explained it a bit better, so
no problem.
Go back.
So guys, yeah, you can go back a bit and allow me to explain.
Speak, I'm going back.
Just explain things a bit. Oh yeah, Jada, I know I've heard the stories of flow, so I don't know. It is just it's
Hold on, is this being recorded? Because I'm not too sure if it is. Y'all see a recording sign?
I'm seeing recording.
You're seeing it?
Yeah.
I don't see it. Anyway.
All right. So what I'm gonna do, guys, I'm not going to, I'm sure Olivia had, raised the points, but what I'll do, I just start back from scratch in terms of what are the key points that you guys remember. Stop here, Olivia.
Yeah, so we're gonna go here.
All right, go back a bit, just a bit more.
Here?
Yeah, all right.
Okay, cool.
So, guys, I'm just going to really and truly speak with regards to what it is that you should pay a lot of attention to. As Olivia explained earlier, we had decided to do to split the portions. So she would be better prepared to speak about translation. I'd be better prepared to speak about transcription. So in terms of the transcription process, what I want you guys to really pay attention to are these 4 things as are outlined here. So the initiation process, as you guys would have gone through, it's basically how that molecule, whether it be the RNA polymerase just for the prokaryotes or the RNA polymerase 2 in the case of the eukaryotes. So how is that going to now mediate the process of transcription? Elongation, of course, as we know, is just adding the incoming nucleotides to the strands to form a continuous band, which would, of course, be the pre-mRNA. And I say pre-mRNA because in the case of the eukaryote cell, which is mostly what we give a lot of attention to, it has to be modified before it can actually be used in the translation process. And termination, of course, now you need to put an end to the process, all right? And you know that you're going to have enzymes involved. These things don't just go on arbitrarily. Next slide, Olivia. So I made mention here about the enzymes that are involved in terms of what it is that for the different species, in terms of the different types of organisms that exist, at least, you have the prokaryotes and you have eukaryotes. In the case of the prokaryotes, you have RNA polymerase doing the vast majority of everything. It will be ensuring that you have separation of the strand. It will have the elongation. It will separate in terms of the histones that are actually there. Because if you remember, DNA is not something that is just laid down like that. It has to be organized, and so it needs those histone molecules to basically wrap it around, and then it's going to form that nucleosome, all right? So we remember that from previous classes. In terms of eukaryotes now, you do have 3 types of RNA polymerase that are used. However, the focus is actually on RNA polymerase 2. And the reason for that is that, so RNA polymerase 1, it is normally used for the synthesis of rRNA. So if we remember ribosomal RNA, it is really and truly used to make ribosomes, all right? RNA polymerase 3, that is actually used in the synthesis of your tRNA molecules. And we know that the tRNA molecules is actually used in translation. So in the translation process, you're going to be using the polymerase, RNA polymerase 3 to make that tRNA so you can use it to actually now make your proteins. It is the RNA polymerase 2 that is normally having that vast majority of the function where the synthesis of the mRNA strand is actually concerned. And so it is really important for that to be intact in the eukaryote. So a deficiency somewhat would lead to, a lot of issues in terms of producing your mRNA molecule and subsequently making proteins, because you need that template before you can actually make your proteins. And I put that the key difference is that the eukaryotic cell is membrane bound. So when you think about the enzymes, if we think about what a prokaryote is, we remember that it lacks membrane bound organelles. So we can remember that the circular DNA is basically just free floating in that cytosol, as well as, chloroplast in the case of certain bacteria and other organelles that are there. So you really actually do not have, I shouldn't say chloroplast, that is, forgive me. It is having structures similar to that. All right, so they have, I think, mesosome, which would be somewhat similar to the mitochondria. And all of these are not membrane-bound organelles. In the case of your eukaryotes, remember that your nucleus has a membrane, your mitochondrion has a membrane, your chloroplasts have a membrane of their own. And what that means is that as you actually go out from one compartment into another, you're going to now have to be protecting your molecules because you're going into perhaps a more foreign environment. And the key difference, as I said, is that you are going to need an enzyme now that can facilitate certain measures to ensure that the viability of the compound is actually maintained. Next slide, Olivia. Next slide. Thank you. All right. And this, of course, is just for us to remember that as the transcription or the whole
Yeah. 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 Yeah.